bone. C-bone, the third hardest tissue, is always being remodeled by responding to pressure by undergoing resorption and to tension by adding more bone. The bony skeleton not only supports the body, but also forms a defensive armor to protect vital organs, such as the brain and spinal cord. Skeletal muscles attach to bones across joints, permitting movement of parts of the body against each other and locomotion of the entire body. Almost all of the body's calcium is stored in the bony skeleton, acting as a reservoir. The calcium can be liberated from the skeleton to maintain the proper blood calcium level. Bone also houses bone marrow in its marrow cavity, which is responsible for hematopoiesis. The outer surface of bone is covered by a soft connective tissue, the two-layered periosteum. The outer fibrous layer is composed of dense irregular collagenous connective tissue. Chapter. The inner cellular layer is osteogenic, housing osteoprogenitor cells, osteogenic cells, some osteoblasts, and, occasionally, osteoclasts. The marrow cavities are lined by endosteum, a thin cellular layer composed of osteoprogenitor cells, osteoblasts, occasionally osteoclasts, and slender connective tissue elements. Bone matrix is composed of inorganic and organic components. Calcium and phosphorus constitute most of the inorganic components, about 65% of the dry weight. Most of the calcium and the phosphorus are present as hydroxyapatite crystals Ko, Po4, 5, O, I that are inserted into the gap regions of and are aligned along the length of type I collagen fibers. The crystals attract water, forming a hydration shell that facilitates ion exchange with the extracellular fluid. Type I collagen, the principal constituent of the organic component, forms approximately 80% to 90% of the organic portion of bone. The bulk of the remaining organic component is in the form of aggrecan composites, whereas osteocalcin, osteopontin, bone sialoproteins, and adhesive glycoproteins complete the organic component of bone matrix. Glycoproteins facilitate the adherence of bone matrix proteins to hydroxyapatite crystals and to integrins present in the plasma membranes of bone cells. Cells of bone. Bone possesses four classes of cells, the first three of which osteoprogenitor cells, osteoblasts, and osteocytes belong to the same cell lineage, whereas the fourth class, osteoclasts, are derived from monocyte precursors, Fig 7.3. Osteoprogenitor cells populate the incanals and the marrow cavities. They proliferate in a cellular layer of the periosteum. They line Haversian, forming more osteoprogenitor cells and osteoblasts when oxygen tension is high, or chondrogenic cells when oxygen tension is low. Osteoblast formation requires the presence of bone morphogenetic proteins and transforming growth factor S. Osteoblasts form a layer of cells that secrete the organic components of bone matrix, as well as the signaling molecules, receptor for activation of nuclear factor kappa B ligand, RANKL, and macrophage colony stimulating factor, MCSF. As the osteoblasts secrete their matrix, they form slender processes that contact the processes of adjacent osteoblasts and form gap junctions with them. As the bone matrix accumulates around the osteoblasts, these cells become incarcerated in the matrix that they formed. They become known as osteocytes, and the space that they occupy in their matrix is known as a lacuna. Osteoblasts not only manufacture bone matrix and become osteocytes, but they also participate in the calcification of the matrix. As bone formation is completed, its outer surface retains a layer of inactive osteoblasts that no longer manufacture bone matrix, but become flattened, and are known as bone lining cells. These cells and osteocytes are separated from the calcified bone matrix by a thin layer of non-calcified matrix, known as osteoid. If the need arises, bone lining cells can be activated to form bone matrix. The cell membranes of osteoblasts possess integrins and parathyroid hormone, PTH, receptors. The former permit osteoblasts to adhere to bone matrix components, and the latter, when they bind PTH, prompt the secretion of RANKL and osteoclast stimulating factor by osteoblasts. RANKL facilitates the transformation of preosteoclasts to osteoclasts, and osteoclast preosteoclasts to osteoclasts, An osteoclast stimulating factor induces osteoclasts to resorb bone. Before they can do that, however, osteoblasts have to remove the osteoid from the bone surface, permitting the osteoclasts to gain access to the calcified bone. Alkaline phosphatase is richly represented in the cell membranes of osteoblasts. When bone is being elaborated, these cells secrete high levels of alkaline phosphatase, elevating the blood level of this enzyme. By evaluating alkaline phosphatase blood levels, it is possible to monitor bone formation. Cells of bone, osteocytes occupy lacunae in bone, and their slender osteocytic processes extend through canaliculi, narrow channels in the calcified bone matrix, to contact the osteocytic processes of adjacent osteocytes, where they form gap junctions with one another and with the processes of osteoblasts, thus facilitating communications among the cells of bone. Extracellular fluid infiltrates the periosteocytic spaces and the canaliculi, providing nutrients and signaling molecules for these cells, and removing waste products and signaling molecules released by these cells. These spaces house more than 1 L of extracellular fluid into which osteocytes can release as much as 20 g of calcium in a short time. Osteoclasts, derived from the mononuclear phagocyte system, are large, 150 gm in diameter, multinucleated cells, N50 nuclei, that resorb bone and possess numerous cell surface receptors, which include osteoclast stimulating factor 1 receptor, calcitonin receptor, and rank. Osteoclasts are activated to resorb bone by osteoblasts that have been stimulated by PTH, and are inhibited by calcitonin that binds to calcitonin receptors on their plasmalemma. MCSF, secreted by osteoblasts, bind to MCSF receptors on osteoclast precursor cells, stimulating them to proliferate and to express cell membrane rank receptors. Simultaneously, osteoblasts express receptors for rank, rankle, allowing osteoclast precursors to bind to osteoblasts. Chapter The rank-rankle interaction induces the trimerization of rank on the surface of the osteoclast precursor cell, activating its adapter molecules to trigger nuclear transcription. The nuclear factors that are produced convert the mononuclear osteoclast precursor into an inactive multinuclear osteoclast, which detaches from the osteoblast. Osteoblasts also manufacture osteoprotogerin, OPG, a ligand that possesses a strong affinity for rankle, blocking its availability for rank and preventing the binding of osteoclast precursor to an osteoblast, preventing osteoclast formation. In the presence of PTH, osteoblasts manufacture more rankle than OPG, and in this manner they facilitate Osteoclastogenesis, development of osteoclasts. Inactive osteoclasts express integrins, allowing these cells to adhere to the bone surface. After osteoblasts remove osteoid from the bone surface, they leave, and their previous location becomes populated by inactive osteoclasts that, by adhering to the bone surface, become active osteoclasts. Shallow depressions located on the bone surface, called Hauschip's lacunae, house these active osteoclasts. Osteoclasts have 4 recognizable regions when resorbing bone. Fig 7.4 The basal zone contains most of the organelles of the osteoclast except for the mitochondria, which preferentially concentrate at the ruffled border. The ruffled border is located at the osteoclast bone interface where resorption occurs. The osteoclast exhibits motile finger-like cytoplasmic extensions whose plasma membrane is thickened to protect the cell as it is resorbing bone, forming a subosteoclastic compartment. The clear zone, the organelle-free region at the periphery of the ruffled border, expresses integrins whose extracellular aspect binds with osteopondin on the bone surface to form a sealing zone, isolating the microenvironment of the subosteoclastic compartment. Intracellularly, the integrin molecules contact actin filaments that form an actin ring. The vesicular zone, the region of the osteoclast located between the basal zone and the ruffled border, is rich in exocytotic and endocytotic vesicles. The former transport cathepsin K, which degrades collagens and other proteins of the bone matrix, into the subosteoclastic compartment, whereas the latter transport degraded bone products into the osteoclast. Mechanism of bone resorption. The acidic environment leaches the inorganic components from the bone matrix, and the dissolved minor ALS enter the osteoclast cytoplasm, where they are exocytosed for delivery into the local capillaries in the vicinity of the basal zone. Osteoclasts secrete cathepsin K into the subosteoclastic compartment to degrade the organic components of the bone matrix. The resultant partially degraded materials are endocytosed by the osteoclasts, where they undergo further degradation before their release at the basal region. C. Osteopetrosis results from a genetic defect in which osteoclasts are formed that are unable to resorb bone because they cannot form a ruffled border. Patients with osteopetrosis present with very dense bones and possibly anemia because of a reduced volume of marrow cavity. These individuals are also susceptible to blindness, deafness, and cranial nerve anomalies as a consequence of narrowing of the foramina through which cranial nerves exit the skull. Chapter